All right, welcome back, everybody. Coming to you live from my house, Delta College, Bio 153 online. So, coming to you here live, streaming out to you guys in the Tri-Cities, out in the Thumb, Northern Michigan. And so, today, our focus is learning about a couple concepts from chapter 26, dealing with urine. As next week you're going to be doing your urinalysis lab, your virtual urinalysis lab. So a very important concept to know in the medical field is this idea of looking at urine and ultimately seeing, well, what types of solutes can be detected in urine, items that would be abnormal. And that's what this picture is kind of showing you here. Here's someone doing a overall analysis of urine. And here's a reagent bottle and a reagent strip. And ultimately what would happen here is the lab personnel would go ahead, have their urine sample, dip the reagent strip into the urine for a couple seconds, take it out, and then actually use this bottle right here, which would match up with the particular solute, if it's glucose or ketones or bilirubin, and ultimately looking at possible color changes on the strips. Typically, if there's no color change, that would be a negative reading. Of course, that's what you always want, is a negative reading. However, if the strip changes color, then we may have a positive reading based off the coloration. Also gives you an idea of the concentration of the solute. So in a typical lab setting for the urinalysis lab that you'd have in the actual classroom, you would have several different unknown specimens of urine, and using these reagent strips to test for the urine, and based off a positive reading of glucose or protein or ketones or bilirubin or whatever it might be, gives you an idea of a possible disease that the patient may have, pathologically speaking. So a very important type of skill set to know going into the medical field is doing a urine test. So we're going to look at a couple slides here that can help you when you eventually do your urine lab. And so we'll take a look at these here next. All right, so the physical characteristics of urine. Here's urine here in a container. Maybe you've all had the experience of peeing into a container like this, going into the hospital, maybe seeing your family physician or even in the ER. And maybe you've done a lab like this in microbiology too, possibly. So some of the couple items here then to consider. One is always do an analysis of looking at the color of the urine. And the color may vary. It may vary from a clear color to maybe even some coloration, pale to deep yellow. And the coloration is based off of the presence of the pigment urochrome. So urochrome is the pigment that results from the body's destruction of hemoglobin. So that's urochrome. You might also consider the transparency when you look at the urine. So are you able to look right through the urine, or is it very cloudy with very low visibility, a lot of turbidity? So transparency is another thing to consider. as well. Even the concept of odor. So, what exactly is the smell of the urine? Is there no smell? Or is there a fruity smell, like you might have with diabetes? Or maybe a putrid smell or a pungent smell, very strong type of smell, which you might find with urine that might be more of a brown coloration with someone with liver disease or cirrhosis of the liver or hepatitis. So odor is something to consider, where you'd actually smell the urine in the lab. And then another thing to consider when it comes to the physical characteristics of urine is taking a look at the pH. So typically the range of pH can be anywhere from about 4.5 to 8. And the variation could be for a variety of reasons. It might be your diet, where there might be an ultimate change. Maybe someone who's been vomiting or even a UTI may alter that pH. So these are considered some of the various physical characteristics of urine. One thing to mention too about the color, usually a healthy color would be more of a pale light coloration of yellow of urine, or known as straw. So straw means more of a light yellow. And sometimes your coloration of urine may change too based off of your diet. If you're eating a lot of asparagus or red beets, there actually will be a change in the color, which is typically normal based off of those various vegetables. A very important concept to consider when we look at urine doing the urinalysis lab is this term right here, which is known as specific gravity. Specific gravity. So this is referring to the ratio of the mass of a substance to the mass of an equal volume of distilled water. So based off of your reading here, if you have a reading between 1.001 to 1.035, this is ultimately dependent upon the solute concentration. So how many solutes is found ultimately in the water. If you simply had a reading of 1, well, that's distilled water. So this is typical to have a reading here in a normal realm for solute concentration, referring to maybe salt or maybe a trace of protein if you have a high protein diet, and etc. You can get the specific gravity reading off a reagent strip, like I mentioned on the first slide. But a very old school way of getting specific gravity would be actually transferring the urine into this glass graduated cylinder and putting the urine in here, filling it pretty close to the top, and then putting in what's known as a hydrometer. So this device here is a hydrometer. They actually used to be made with mercury back in the day, and you would put it in the urine, you would go ahead and spin it, you'd spin the device, and you'd wait till it would actually settle, and then based off of that, you could actually read off the values of specific gravity on this hydrometer device based off the meniscus reading of the urine, and then you'd get that actual reading of the specific gravity. It's also known as a urometer, so that's the same type of device. This is something sometimes you would even utilize if you were brewing beer or wine. You would use a hydrometer reading too. But we don't use these anymore because if you drop it, then you have mercury in the room, and then, obviously that's not safe. So we'll be taking specific gravity using reagent strips. Well, what exactly is in the urine? So we look here at the chemical composition of urine. Again, this is considered, items you would find in a normal situation. So approximately, when we look at urine, about 95% of urine is made up of water and 5% solutes. And when we're considering the solutes, again, it could be things like such as there could be some nitrogenous waste products. That's pretty normal. So urea, again, this is from protein metabolism, or uric acid, or even creatine. So creatine, again, this is a product formed from creatine when we think of muscles. So these are nitrogenous waste products. Other normal solutes you would find in the urine, typically speaking, would be some sodium, potassium, calcium, magnesium, bicarbonate. So this gives you an idea here offhand. And sometimes you might even see a trace of protein, possibly if someone has a high protein diet, which is fairly normal. So those are some of the things you would find as an example. Maybe even some hydrogen ions as well. But any abnormal high concentration of a solute potentially may indicate someone has a pathology or disease. So hence the importance of doing a urinalysis sample when you're in the hospital and the physician's trying to figure out what's wrong with the patient. This table here gives you an idea if you find some of these particular solutes in the urine in high concentrations that someone may have a particular type of disease. So for example, if someone were to have significant high amounts of glucose in the urine, this may indicate the patient is diabetic. So that may indicate, well, they may have type 1 or type 2 diabetes, very possible. Proteins, if someone were to have a high amount of proteins, from a pathological standpoint, it could mean they have heart failure. It could mean maybe a very high blood pressure or even renal disease, different forms of renal disease. For ketones, if someone were to have high ketones, this would be an example of someone maybe in starvation mode, obviously not good, or they're not properly treating their diabetes. So they have the ketone breath is usually a possibility there. If per se someone had high levels of hemoglobin, so an abnormal amount, maybe because of a blood transfusion reaction or some form of anemia or even severe burns. The possibility of high biopigments, so possible high levels of bilirubin, would be a possibility of liver disease for the patient, either hepatitis or cirrhosis, or even obstruction of the bile duct from the liver or gallbladder. This one here, erythrocytes, red blood cells. If someone would to have high elevated amounts of red blood cells in the urine, it could mean a bleeding urinary tract, could be severe trauma to the kidney, kidney stones, infection, are all possibilities. And the last one, if someone were to have elevated amounts of white blood cells, leukocytes, possibility of a UTI. More likely than not, the patient may have a urinary tract infection. So very important to make sure to know that. So these are all possibilities here, and definitely important to review these slides ultimately when you perform the lab. So later this week, I'll have data sheets already with prepared data. And ultimately what I want you to do is to go ahead and identify what disease the patient might have based off of the data I provide you. And when it comes to studying of your quiz over the urinalysis lab, make sure you study these slides. Make sure you study these slides. So definitely make sure to do that. Okay, that's very, very important. Along with reviewing the lab itself, and your course packet is critical too. And that's all I have for you today.